and thank you, Ricardo, for invitation to today's webinar. My name is Kat Ginda-Makela, and I would like to introduce you to our company, Applied Cells, and to the technology we have developed, a Mars platform. Our company is based in California and Silicon Valley, and about 4 years ago, we have launched the first Mars system, providing a new approach to sample preparation with a more gentle, more efficient and rapid solution for easy sample preparation and for cell manufacturing. So we do this by offering our customers two technologies, new acoustic separation and in-flow immunomagnetic cell separation. Today, I will focus on the in-flow immunomagnetic cell separation that has no need for dedicated columns. It relies on a classical immunomagnetic labeling, where magnetically labeled cells of interest are captured in the magnetic field and unbound cells are collected in the negative fraction. What's very unique about our approach is the fact that we don't use dedicated columns or any kind of matrix. The cells flow through the tube surrounded by magnet, and magnetically labeled cells are captured along the tube, while all the unbound cells flow through. Next, the magnet is removed and labeled positive cells released in the positive tubes. So here's the little video showing how it works in practice. This is the example instrument with 3 magnetic modules. You can run 3 samples because the magnetic modules are identical. In each of the modules, you can see cell separation channel and the magnet. So in the first step of the process, the door closes and sample is loaded with magnetically labeled cells. On this particular example, it's a peripheral blood with cells, with selected cells magnetically labeled. And what happens as the sample flows through the channel, magnetically labeled cells stay along the tube, while all the unbound cells are flowing through and they are captured in the negative fraction. Then the cells are rinsed, the door opens, and the little arm shakes the tube in order to release the magnetically labeled cells to the positive fraction. Subsequently, the door closes and the cycle continues. So we can do this process multiple times, as many times as necessary to separate all the cells of interest. A single loading of the tube captures between 100 and 500 million cells. So the capacity is significant, but it gives us the flexibility to increase the volume of the separated material. So the lack of dedicated column gives our platform several unique advantages. One, because we don't use columns, we don't observe extensive trapping of non-specific particles, which greatly increases the purity. 2, no cells of interest are trapped on the column, which means that we can achieve much higher recoveries. 3, no need to purchase columns significantly reduces the cost of associated experiments. And because we collect both positive and negative fractions, we can very easily do both positive or negative selections. And finally, my favorite feature about the system is unlimited capacity, because Mars bar doesn't use columns. And as I just demonstrated, there is no limit to the number of cells that can be captured, because the door opens and closes, the capturing cycle continues. So the magnetic modules were designed to handle a wide variety of sample types, including whole peripheral blood, cord blood, bone marrow, apheresis, PBMCs, and many others. We can run low volumes, starting with 0.5 milliliter, as well as sample bags, which gives us a flexibility in the experimental design. And here are the core applications that we focus on. Today, I would like to talk more about target cell isolation. I will also mention cell therapy and debris removal for genomics or flow sorting. So to start with target cell isolation, one of main applications where we bring a lot of value is CD138 plasma cell isolation, CD34 stem cell isolation, And we also have some other examples of other immune cell isolations as well as cancer cell isolation. So what's unique about Mars is that the platform simplifies the cell isolation process through its very fast and easy workflows. It improves purity while maintaining very high recovery of the cells. Our instrument uses the automated 1 to 3 pass enrichment process, and it involves minimal hands-on time. In practice, it means that you can use single module to separate your cells, but if you want to increase the purity without sacrificing recovery, you can set it up to run second and third enrichment as a totally walk-away solution and achieve very high efficiency of your process. So our technology has the ability to directly isolate target cells, including those that are present in very small starting percentage from, for example, fresh bone marrow samples. And this is especially important in the multiple myeloma research and in diagnostic space, where advanced diagnostic techniques, including FISH or flow cytometry or even genomics, are required for good timely disease identification. However, the successful and reliable detection largely relies on the efficient enrichment of CD138 plasma cells. So in this particular case study, we have isolated plasma cells starting from a very low starting percentage, less than 0.5%, and enriching the sample through the two-step pass to 95% of purity, while maintaining very high 88% of recovery. And this is the other example. Here we used a spike-in experiment, and we have enriched U266 cells at the starting percentage, which was a bit higher, about 5%, and reaching it to 91% with a single pass separation. The recovery, as always, was maintained very high, almost 90%. And since the rising interest in multiple myeloma research, I would like to mention here that we have recently opened a CellQuest grant, which is open to all clinical research scientists with a special interest in multiple myeloma and cell therapy development applications. This initiative includes a 6-month placement of the company's proprietary Mars-Bar cell separation instrument, along with reagents and consumables, training, application development support, and continuous support, in total worth up to $30,000. So the program aims to support acceleration of our understanding of multiple myeloma. And I would like to encourage you to check our website, and if it's of interest, of course, I would like to encourage you to apply. But coming back to the presentation, another application where Mars is proving to be the game changer is isolating CD34 positive cells. In this example, this is a case study where our user has isolated CD34 positive cells from cord blood mononuclear cells, an assay that is crucial for research in hematopoietic stem cell biology or therapeutic advances. By utilizing the automated CD34 enrichment process, the workflow has enhanced the purity starting from 0.18 to almost 95%, while ensuring the reproducibility, the high recovery of 83%, and high viability of isolated cells. And what's very important, in this particular case, using Mars has decreased the assay cost by 60% for our user, as compared to the current method he has been using. And it also significantly cut down the experiment time by approximately 50% for 50 milliliter input sample. And this is because we have reduced the number of steps necessary for conduct. It was reduced from 8 steps to 4. The inflow technology introduces the flexibility and the efficiency in the workflow optimization. And here I'm presenting the simplified workflow, a protocol, which begins with a very straightforward single or two steps labeling of cells of interest, then dilution, which is followed by the automated immunomagnetic separation process. Here is the example of the isolation of CD34 positive cells from mobilized blood. Single cell enrichment of the single pass enrichment of CD34 HSC cells from baseline of about 2% led to a purity of almost 90% with recovery above 94%. And the following two slides show a range of examples for immune cell isolation. From T cells through monocyte, B cells, we consistently achieve 90% and above purities and recoveries, and we we reduce the need for extensive pipetting or for multiple centrifugation steps. And here is the example of monocytes and B cells enrichment. One of applications that I would like to mention today is cell therapy development. Here we offer research use only solutions for easy assay optimization and development. And currently we are also partnering with our users in GMP solutions for labs that have the requirement to work in the clean room. All the processes are fast, fully automated, and they are optimized to provide economical solutions. So in the CGT cell manufacturing or cell therapy development, Mars and our solution falls into immunomagnetic cell isolation step. Here we not only provide the instrument that I will talk a bit more about, but also reagents. We offer direct conjugate in our Mars Ingenuity line, which supports researchers both on the RUO and GMP side. Additionally, our software is designed to be 21 CFR 11 compliant with controlled user access or encoded protocols, so it is ready to use in cell therapy development. And as always, we focus on making the magnetic cell separation workflow as easy as possible. In this example graphic, you can see a 5-step cell separation from the leukopak, which begins with magnetically labeled target cells. Then the bag is diluted, it's installed on the dedicated rack. The user is selecting the protocol, presses run, and is ready to collect the enriched target cells. Overall, the workflow with Mars provides a simplified solution which saves time. There's no need for centrifugation or extensive sample processing, which reduces cell loss. And the overall benefit result is a cost-saving as well as time-saving solution. And the example data that was produced by our user that I'm presenting here is CD4 CD8 T-cell isolation. In that particular experiment, what is worth mentioning is the very high recovery of over 92%, is maintained viability of the cells, and very fast and easy workflow to quickly get to the enriched cells population. Another example is TCR gamma delta purification through TCR alpha beta depletion. In this case, we have been compared against a competitive solution. And while the competitive solution provided very good results, our process turned out to be much faster and resulted in almost 100% purity of the TCR gamma delta cells. We have also maintained very high recovery of over 90%. Another example, NK cell separation, we are often asked by our users whether we support the NK CD56 positive, CD3 negative enrichment. Yes, we do. So in a 2-step separation where we first deplete CD3 positive cells and then enrich CD56 positive cells, we achieve over 91% purity for the NK cells enrichment, and the recovery is maintained on the high level of about 76%. So here is the range of reagents, and here is the way we support cell therapy development from end to end. Starting with a Mars bar flex system that supports a small-scale assay optimization, we support the discovery phase. Users can run up to 3 samples at the same time, 1 or 2. They can run different volumes, and they can quickly test different conditions to initially develop their protocols. When ready, they can go to upscaling using our RUO large-scale Mars bar system. This allows them to quickly adopt the protocols and optimize the protocols to be ready for transition to the clean room. And when ready, using our ISO-certified Mars bar and dedicated reagents, they will be able to move their manufacturing to the clean room in an established manner and with a reliable protocol. Now, the final part of my talk today focuses on the debris removal using the acoustic technology for efficient cell or nuclei separation from debris and dead cells, while I'm maintaining high integrity and high viability of the purified cells or nuclei. So the acoustic separation is a new approach that separates particles based on the differences in their sizes, physical or mechanical parameters that include buoyancy or their density. So how do we do it? Cell or nuclei suspension that contains live cells, dead cells, and cellular debris passes through microfluidic channel in a laminar flow with the fresh buffer at the center stream. When the acoustic field is on, live cells will move to the fresh buffer in the middle stream from the original suspension, while dead cells and debris will remain in the outer stream of the flow. The cells or nuclei of interest are then recovered in the sample tube, while the cell debris and lysed red blood cells will be collected in the waste tube. So acoustic separation allows for a label-free cell isolation, concentration, and filtering bioparticles. And additionally, the applied acoustic waves are in a similar frequency and a power range as those that are used in ultrasonic imaging, for example, and they have been tailored to avoid damage to particles and cells. So because there is no FICO or no gradient or centrifugation, the Mars process is much more gentle and it's much faster, thus maintaining high cell viability. Conventional methods for debris removal and commercially available cleanup protocols involve the use of many centrifugation steps, magnetic separation, or fast sorting in some cases. And the process can be very time-consuming. It can also be costly. Often cells or nuclei of interest are affected by the treatment. What's more, centrifugation methods often don't succeed in removing the debris efficiently. And in contrast, Mars acoustic wash is a gentle single-step process. So cells or nuclei suspension after straining is passed through the acoustic chip and is separated in the acoustic field in a fast, several minutes long process. The flow rate is on average 1 milliliter per minute. So typically the sample is resuspended in 1 to 2 milliliters, is ready within several minutes. It's ready for the downstream application. This ensures high quality of the cells and nuclei, as they are not overprocessed, they are clean, and the debris is usually significantly reduced. So here is the example data where we compared cell isolation from lung solid tumor biopsy. We isolated cells using Mars in parallel with centrifugation. As you can see, centrifugation did not succeed, was not very efficient in dead cell and debris removal. And you can see, especially here in the dot plot, that Mars debris removal was very high, over 90%, and cell purity was over 40%. In this experiment, we confirmed that the acoustic separation does not preferentially enrich a specific subpopulation of white blood cells. In fact, all the analyzed white blood cell subpopulations were enriched with a recovery higher than 92%, and it was much higher than the three-step centrifugation method that we also used as a reference. So I would like to mention a bit about our instrument. Mars platform consists of a range of instruments that provide a flexible solution to meet very specific experimental needs. We offer different systems depending on the required applications. So our SP system offers only acoustic, untouched cell separation modules. These are very useful for debris removal and unlabeled cell or nuclear enrichment. Mars bar comes in 3 versions. Research use only small-scale version for cell therapy discovery phase or multiple myeloma and CD34 research. Large-scale version RUO for optimizing protocols for cell therapy development. And finally, large-scale version, a version that is ISO certified to be used in the clean room for cell therapy development. And finally, Mars CS. That system has both Mars applied cells technologies. It consists of 2 acoustic modules for untouched cell enrichment and 1 magnetic modules for immunomagnetic isolation. We typically recommend this system for more versatile applications or more complex assays that may require first debris removal and then magnetic enrichment. In all cases, all our systems can be sterilized and can be used in a biosafety hood. As you can see here, the tablet can be detached and can be kept outside, for example, for remote control. All fluidic components can also be easily replaced with new ones, sterile ones. And the replacement is very easy. It literally takes 5 minutes. All the fluidic components are disposable and they are sterilizable. Now, the system has also built-in redundancy. And so in the unlikely event of clogging, 1 module will pause, but the 2 others will continue running. And this means that the process does not stop, the precious sample is not lost, and we additionally provide regular and deep clean programs for more efficient system maintenance. The tablet itself offers a full touch GUI, which can be remotely updated by our engineering team. And during the operations, the system also offers audiovisual guidance, leading the user step by step through the process. So in the final part of this presentation, I would like to also mention our reagents. The system is open, and we would like our customers to have the opportunity to adopt the system for their unique applications. But we also offer reagents that are, of course, supported by protocols that have been specifically optimized for Mars platform. They offer fast isolation, flexible protocols, and they constitute an economical solution. First of our reagent lines, Mars Mag line, utilizes PE anti-PE binding. We provide PE conjugated beads and a range of anti-PE conjugated antibodies targeting commonly isolated biomarkers. The Mars Mag Premium line, developed in collaboration with stem cell technologies, offers ready-to-use cocktails for isolation of cells, including plasma, CD138 cells, CD34 and NK cells, etc. And finally, Mars Ingenuity line. This is a line developed for collaboration in the cell therapy development, where we are offering direct conjugates that come in RUO or GMP versions for isolation of most commonly used cell therapy type of cell populations. So thank you very much. And I hope that Mars can also be a solution for you. Please reach out to either representative Axela teams or directly to us if you have any further questions. Thank you.
Thank you. Thank you. Maybe I have a question about the reagents.
Yeah.
So this